Hello everyone and welcome to Chapter 7 of Mastering Biology. Today we'll be talking about Concept 1 in this chapter, Catabolic pathways yield energy by oxidizing organic fuels. What we'll be doing today is going over the basic concepts and principles pertaining to how cells are able to produce the energy that they need to keep living and to sustain their various processes. Then we'll have a brief preview of the exact steps and processes that make up cellular respiration, which will be covered in more depth in Concept 7.2, 7.3, 7.4, 7.5, and 7.6. Catabolic pathways involving electron transfer are central processes to cellular respiration. Bit of a mouthful. To kind of break that down a bit, what we're talking about is cells breaking down molecules and then using the electron movement of those reactions to get the energy that they need. Here are some key concepts and words to know about this process of getting energy from the molecules that cells take in. That first bullet point is pretty important and hopefully pretty obvious. The breakdown of organic molecules is exergonic. It's releasing energy. This makes sense. We eat food, we break it down, it gives us the energy that we need to keep our cells going. When we talk about respiration, we're talking about the process of breaking down those molecules to get energy from them. And there are kind of three big classifications of respiration that you should probably know. The most important, and what we'll spend the most amount of time on, is aerobic respiration. So that's basically you're breaking down molecules, you're using lots and lots of oxygen, and you're getting the ATP, the energy that you need from these molecules, out. In anaerobic respiration, you're doing basically the same thing, but you have no oxygen to play with. It's just like upping the difficulty level a little bit. Finally, we have fermentation, which is where you're degrading sugars without oxygen to get the ATP that you need. When we say cellular respiration, we're talking technically about aerobic and anaerobic processes. But a lot of times when someone says cellular respiration, what they really mean is aerobic respiration, especially when it comes to the steps and the concepts and some of the key players. If I say cellular respiration, or if someone in class says cellular respiration, they're probably talking specifically about aerobic respiration. Another kind of key caveat to know, during this presentation we're going to be talking a lot about glucose as kind of the basic thing that is being broken down to release energy. Don't take away from this that glucose is the only fuel that's being used. That's the only food. Your body takes in tons of different things, carbohydrates, fats, proteins, and we use almost all of them in one way or another for fuel. But it's kind of hard and confusing. So rather than looking at how we manipulate fats and manipulate proteins and handle different types of carbohydrates, it's easier to just say glucose, our good friend C6H12O6. Right there below that bullet point, you can see the general reaction that you have with glucose and oxygen reacting to produce carbon dioxide, water, and the ATP that we need for energy. So to hit back on that first point that I was making earlier, the transfer of electrons during chemical reactions releases energy that's stored in these molecules. So the movement of electrons is really what's driving this process. The energy that we get from electron movement is, once it's passed through this whole cycle, kind of synthesized and manipulated to get us ATP, produced from a variety of different things. The basic chemistry behind this comes through redox reactions. Redox is of course named for the red ox that discovered them in the 19th century. It was really one of the greatest animal contributions that we've had in science. Just kidding, of course, it's a portmanteau of reduction and oxidation. These reactions are basically just transferring electrons between reactants. So oxidation is when a substance is losing electrons, and reduction is when a substance is gaining electrons. It's kind of confusing, but there's a pretty easy way to remember it. Oil rig. It's a handy little acronym. Oxidation is losing electrons, reduction is gaining electrons. This all can get kind of complicated when we talk about different agents and kind of wackier compounds, but just if you can remember, oil rig, that you're oxidizing when you lose electrons and reducing when you gain electrons, then you're pretty much good to go. Let's look at some examples. In the first line, you have a combination of sodium and chloride. When we react those together, you get sodium with a +1 charge and chloride with a -1 charge. So what happened is the chloride gained an electron, so it was reduced. It's kind of confusing because it's getting the electron, but also being reduced. Think of it as charge is what you're concerned about with the names. So it dropped from a charge of 0 to a charge of -1 That was a reduction. Conversely, sodium went from having no charge to having a +1 charge. It was oxidized because it lost an electron. The general form that you see on the bottom line kind of shows this. So you have an electron shifting from one of the reactants to the other. The one that loses that electron is what's being oxidized. The one that gains the electron is being reduced. In these reactions, the reactant that is donating the electron, that is losing it, we call that the reducing agent because it's enabling the reduction of the other one. Conversely, and hopefully obviously, the thing that's accepting that electron, that's gaining that negative charge, is the oxidizing agent. Now things get a little trickier. You can have redox reactions that aren't using ionic compounds. With ions, it's pretty easy to see what's being reduced, what's being oxidized, because you have electrons fully jumping from one reactant to another. It's pretty cut and dry, it's pretty easy. But you can also have redox with covalent bonds, where something has a tighter or a looser pull on a certain electron. Unfortunately for us, these covalent redox reactions are mostly what you get in terms of cellular respiration and the processes in there. The example they give here is methane and oxygen gas. So if you look at this figure here, you see that methane starts out with a pretty even sharing of electrons between the carbon and the hydrogen, and the same goes for the oxygen gas. The electrons are not being pulled one way or the other. When you react these, you produce carbon dioxide, you produce water, and you produce energy. And in that process, you shift where those electrons are just because of the differing electronegativities of the different elements at play. So carbon is oxidized because it loses pull on those electrons. When it shifts from being with hydrogen, where it's slightly more electronegative, to oxygen, which is way more electronegative than carbon is, that carbon loses pull on those electrons, so it's oxidized. Flip side of that is water. It goes from oxygen gas, which it's just oxygen, so they're sharing evenly, to sharing with hydrogen, which is way less electronegative than that oxygen is. So oxygen is reduced because it gains electrons. Not all the way, but it gains a stronger pull on them. Redox reactions that have this sort of phenomenon occur, where electrons are moved closer to electronegative atoms. This produces a release in chemical energy that we can put to work, and that's kind of what cellular respiration is all about. Okay, so let's dive right in. During cellular respiration, fuel, which I know we're going to use glucose a lot, but it really can be a number of things, but fuel is oxidized and oxygen is reduced. That makes oxygen an oxidizing agent, which is what you're going to see come up quite a bit. Now that fuel can be a variety of different molecules, but really we want things with a lot of hydrogen. Carbohydrates work well for this, fats work well, because those hydrogens, when they get transferred to oxygen, are going to produce a lot of energy. They're going to release a lot that we can then convert in an ATP synthesis setting. If you look at the line right there, you see the reaction of glucose with the handy redox labels. So when we take glucose and we convert it into carbon dioxide, it is oxidized because it is losing a lot of electrons with all the hydrogens that it doesn't have anymore. Flip side of that is the oxygen, which is reduced as it becomes water and takes in all those hydrogens. Conveniently enough, a lot of energy is put out by this process, which is what we then use to synthesize ATP. In cellular respiration, glucose, or whatever fuel you're using, is broken down in a series of steps, right? We don't just jump in and take care of the whole thing for reasons we'll get into, but we go through a set process that slowly takes out that potential energy in the form of electrons. The electrons that we find in glucose or whatever fuel is being used are typically first transferred with NAD+, the real MVP of cellular respiration. NAD+ is a coenzyme. So if we remember from last chapter, that's something that's just helping an enzyme perform its fundamental function. What NAD+ does is it accepts these electrons really, really well. So it goes into a molecule and it grabs them and it can move them wherever those electrons need to go. When NAD+ goes to work, at least in a glucose setting, it typically takes 2 electrons and 1 proton to form NADH. this new compound, this new molecule, is basically a big unit of stored energy, because we can put that in the cellular respiration process, and it will then push those electrons out. They go through a whole transfer line, and that feeds the energy to synthesize ATP. For NAD+ to become NADH, to pick up those 2 electrons and the 1 hydrogen ion, we need a specific enzyme called dehydrogenase, which, rolls right off the tongue. Here you have a couple pictures to kind of go along with this. On the left you see NAD+ in its format, and on the right you see a picture of how we would take some fuel and use NAD+ to oxidize NADH and get those electrons that we need. As I was kind of alluding to before, when NADH has the electrons that it needs, it passes them to the electron transport chain. How this chain works is the electrons just get moved over and over again to increasingly electronegative things through a bunch of redox reactions. The basic idea here is that if you just let them go to oxygen, right? You had a bunch of electrons and then they shifted over to oxygen, then you would get one big release of energy. That's not that helpful, because there's only so much energy you need at any one point, and you want it to last a long time. So instead, those electrons just get pushed down the chain, yielding a little bit of energy each time. That energy is then converted to regenerate ATP. Here's a pretty helpful graphic to kind of show that point. So the option you would see on the left is just one big jump, and it gives you a lot of energy, and then you're done. On the right, you have what we actually use, which is a series of controlled reactions going down a chain, producing the energy that we need to make a consistent amount of ATP. A good way to think about this, which they used in the textbook, was like gasoline in a car tank, right? You could just throw a match in there and it would just blow up and it would look awesome, and you would probably die, but it would look really cool, and then you'd be done. Like, you couldn't drive with that. Instead, we have a system with an engine where we slowly burn gasoline, lots of little tiny explosions that let us drive where we want to go. Alright, let's begin our preview of the stages of cellular respiration. There are 3 of them. We open with glycolysis. Here's where we take glucose and we break it into 2 pyruvate molecules, and we get a little bit of ATP out of this, but not a whole lot. In the next cycle, we have The next step is the citric acid cycle, where we take that pyruvate and we break it down a little more and we manipulate it and get a little bit of ATP. And then we finish with oxidative phosphorylation, which is where we really take all the energy that we've pulled out in the form of electrons in the first 2 steps, and we put it to use making ATP. Here is a lovely video, courtesy of the finest animation available in 2007 from our friends at Pearson.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
All right, let's go through each of these steps in a graphic format. So we start out with glucose, and then we use enzymes and we use NAD+ to pull off some electrons and split the molecule in half into 2 molecules of pyruvate. Now, as you can tell, this is occurring outside the mitochondria. It's just happening in the cytosol, kind of floating around. Next up, and happening inside the mitochondrion, we have the citric acid cycle, which is also known as the Krebs cycle. What happens here is the pyruvate that was produced in glycolysis is broken down further. More of its electrons get taken away by NAD+ and by FAD+ and transferred towards that electron transfer chain. Now, you see there, the FAD+ isn't something we've talked about. NAD+ and FAD+ are very similar. Think like a Ben Affleck / Casey Affleck split, like serving basically the same function in society, just with slight differences, and one being Batman and the other not. Most gets done here with oxidative phosphorylation. So here, we've taken all of these electrons, and now we put them in that transfer chain. They start dropping down the ladder, releasing energy as they oxidize, and then eventually creating the conditions that are needed to synthesize ATP. Oxidative phosphorylation accounts for about 90% of the ATP generated in this whole process in cellular respiration. How it works is basically you transfer inorganic phosphates to ADP to produce ATP, and the energy that you need to do that is given out by the electrons moving down the chain. There are, of course, smaller amounts of ATP formed in glycolysis and in the citric acid cycle by substrate level phosphorylation, just happening on a much smaller scale and with slightly different conditions, which later concept chapters cover. Oxidative phosphorylation accounts for about 90% of the ATP generated in this whole process in cellular respiration. How it works is basically you transfer inorganic phosphates to ADP to produce ATP. And the energy that you need to do that is given out by the electrons moving down the chain. There are, of course, smaller amounts of ATP formed in glycolysis and in the citric acid cycle by substrate level phosphorylation, just happening on a much smaller scale and with slightly different conditions, which later concept chapters cover. If you go into this whole process with one molecule of glucose, ideally you wind up with 32 molecules of ATP, which is a pretty good yield. Here's a picture of how we produce ATP and ADP. It goes into not a whole lot of detail and is not particularly helpful, but you see the basics of how an enzyme is taking a phosphate group, taking ADP, and using it to produce ATP. The Pearson slideshow inexplicably just ended on that picture, which is not too helpful. So here's a very broad review of the important things that we covered here. So our big overarching idea is that cells produce energy by breaking down molecules and manipulating electrons that get moved around in the process. So this is accomplished through aerobic and anaerobic processes. Aerobic processes have oxygen to play with, anaerobic ones don't. When aerobic respiration takes place, when cellular respiration is going on, it happens in 3 stages that we've talked about. We have glycolysis, we have the citric acid cycle, or as that guy would call it, the Krebs cycle, since he was the one who came up with it, and then oxidative phosphorylation. My personal MVP in all of this is NAD+. That's the molecule, the coenzyme, that's going in and getting the electrons and moving them out of whatever fuel we're using and putting them toward the electron transfer chain. Finally, the big overall goal of this is producing ATP that we will put to great use, powering ourselves and staying alive. Thank you all for listening, really appreciate it, and hope you have a great rest of 4th period.